Thanks, Dan, and thanks for the opportunity to come and talk this afternoon. I'm going to be talking about C4 grasses, and this is a short talk, so introducing C4 really rapidly. It takes some fairly fast talking. I'll do my best. Plants, as many of you probably know, use one of two photosynthetic pathways. The most common is what's known as the C3 pathway. That's because the earliest detectable product of photosynthesis is a 3-carbon compound, phosphoglycerate. This is clearly ancestral, and the central enzyme involved is Rubisco, and it's just kind of pretty, so I put it there. I'm going to mention it once or twice. The alternative, which is much less common, is C4. C4 plants have the C3 pathway, but they've added this sort of turbocharger onto the front of it to improve the efficiency of the pathway and the efficiency of Rubisco. So in terms of familiar plants, rice uses the C3 pathway. Maize, or corn, or zea, whatever you want to call it, is C4. In addition, many of the major biofuels crops, things like sugarcane or switchgrass, are also C4 plants. And C4 plants are much more efficient. They make much more efficient use of light, of water, of nitrogen, and hence they're a lot more productive. This means that they're really attractive. The pathway is a really attractive target for engineering into C3 crops or C3 biofuels plants to make them more productive, particularly in a warming world where C4 is much more effective. That's if we knew the regulators of C4. And one of the messages from today is we don't know the regulators. C4 is a bit complex. All of the ancestral C3 biochemistry happens in a single cell. All of photosynthesis operates within one cell, whereas C4 takes that whole C3 pathway, crams it into a cell around the vascular tissue, and then it captures carbon from the air in an outer cell known as a bundle sheath cell, known as a mesophyll cell. Mesophyll, of course, is simply Greek for middle of the leaf, so it doesn't really tell you anything very much. So looking at this picture here from a standard textbook, there's a mesophyll cell here, right adjacent to a bundle sheath cell. The carbon dioxide is picked up here, turned into bicarbonate, then attached to a 3-carbon compound so that the first detectable product of photosynthesis is a 4-carbon compound, hence the name C4. That then gets moved into the adjoining cell where that carbon dioxide is taken off, handed to ribulose 1,5-bisphosphate and Rubisco. So now you've got a perfectly good C3 pathway going on. It's simply sequestered in a single cell type. Once you've taken that carbon dioxide off, then it leaves you with this 3-carbon thing that can then get shunted back out. So you've got things moving back and forth all the time, requiring an awful lot of transport, an awful lot of communication between cells. In terms of the regulation of C4, we know a little bit. We know there is no master switch. The crossing studies and many, many studies have failed to find anything that even looks like a single gene change that would trigger this whole pathway. The C3 enzymes, the things that drive that set of reactions I just described, are normal housekeeping genes. There's nothing unusual about them. So we're not looking for something that's radically different. The regulation of vein spacing is completely unknown. So what makes those cells, that creates that proximity of cells is quite unclear. So there have been a number of attempts to identify genes that control C4. And one obvious one that was taken up by my colleague Tom Brutnell, who's a co-author in this talk, is to compare the leaf transcriptome of a C4 plant, that's maize, with that of a C3 plant, that's rice. And this takes advantage of the developmental gradient along the grass leaf. So that what you have here is a maize leaf laid out sideways. The bottom of the leaf is over here, and it's undifferentiated, not photosynthesizing, and the other end is fully differentiated and is photosynthesizing quite happily, which means that then the photosynthetic machinery gets put together along the leaf so that if you then look at the transcriptome in various segments going up the leaf, you can get a hint of what kinds of changes happen in the development of the photosynthetic pathway. So this is a maize leaf, and then that can be compared to a rice leaf, and there's a bit of juggling that has to go on, a lot of computational work that has to happen to try to figure out points of correspondence. But once those points of correspondence are put together, it's possible to then look at differentially expressed genes along the gradients of both the C3 and the C4 leaf. And when the Brutnell lab did this, this is all published in this paper here in Nature Biotechnology, when they did this experiment, what they found, among other things, among quite a few other differentially expressed things, were 118 differentially expressed transcription factors. So that's a nice start, but it's kind of a big number. There's also a problem with this sort of comparison in that these two organisms and their difference in photosynthetic pathways confounded with phylogeny. You're looking at at least 110 million years of evolution, separating them, that is, 55 million years on one branch to rice and 55 million years on the other branch to maize. So that's an awful lot of time to accumulate mutations that have nothing to do with the photosynthetic pathway. So the solution would be to find species that are closely related to, find C3 species that are more closely related to maize than to rice, thus sort of cutting down this massive phylogenetic problem. In addition, you can take advantage of the fact that the C4 pathway, complicated though it is, has arisen in parallel multiple times in evolution. There are at least 65 origins within the angiosperms. This is from a paper by Rowan Sage. And then there are between 22 and 24, depending on how you do the analysis, multiple origins within the grasses alone. So that we should be able to find other grasses that have invented C4 independently of maize. And in fact, you can do that. You can find additional C3s and additional C4 origins within the grass subfamily Panacoideae. The deepest divergence in that family dates around 25 million years, and of course then most of the comparisons are going to be substantially less than that. So that's basically what we did. So just showing you in a little more detail, I just talked about the C3 rice and the C4 maize. There are a few other genomes available: sorghum, switchgrass, and Ceteria viridis, the green millet. Those are all C4, as you can see by the orange coloring. And what we did as a collaboration between my lab and Tom's lab was to sort of fill in the panacoid grasses, fill in the set of genomes so that we now have a couple of additional C4s and a couple of C3s that are far more closely related than rice is. So getting our way out of this sort of phylogenetic confusion. So we put in a CSP proposal. We've been sequencing, the JGI has been sequencing an awful lot of these data, and also have done leaf transcriptomes of each of these species. So we have both the genome and the transcriptomes. The transcriptome data have been collected. The libraries are made. I believe they are all sequenced, but only the one in Dichanthelium, that is this C3 species right here, only the Dichanthelium transcriptome has been analyzed yet. The other genomes are either in progress, assembled. Dichanthelium has a paper in review, and the other couple are on their way. So let me just give you a couple of vignettes of the kinds of things that we're able to spot from this sort of analysis by having a sort of phylogenetically controlled sample. The first I want to mention is some work by Pu Huang, who's a postdoc originally in my lab, now in Tom's lab. And he's used the phylogeny to identify genes with elevated rates of non-synonymous substitutions. So these are genes that are presumably under some sort of selection. And the power of this approach depends on having at least two independent origins of C4. So he's looking at things that are not only under selection, but under selection in two separate C4 lineages. And what he finds is 88 genes that fit that criterion. Using that, he can look at all the core C4 enzymes, the biochemistry that's very well known. Some of them are under selection, some are not. All the transporters, the things that move the things back and forth between those two cell types, are under selection. And he's also identified some genes previously unconnected to C4. So it's pointing out some new targets that we wouldn't have spotted otherwise. Another observation that comes out of this sort of data, some work by in James Schnabel's lab, the Rubisco small subunit. I mentioned this major enzyme, and what I didn't tell you is that the protein is encoded by the large subunit. It comes from a gene in the chloroplast, and the small subunit from a gene in the nucleus. And in most photosynthetic organisms, the small subunits are in tandem arrays. The copy number is relatively high in most angiosperms, and indeed all the ones that we've sequenced, including Ceteria and paspalum, which are C4s. But James has found that the copy number was reduced to 1 in sorghum and 2 in maize, and in addition there was a change in the genomic location. Now, if we didn't have those independent C4 comparisons, we would have said, oh yes, this is correlated with C4, if we were just comparing maize and rice. But of course, in this case, we see that the change is in fact unique to that particular lineage. It's not, in fact, a C4 correlation. It's not required for C4 function. The third little vignette I just want to mention is looking at Dichanthelium oligosanthes. This is a C3 plant where we have the genome and the transcriptomes. This is work led by Tony Studer. The first thought is, given its position in the phylogeny, is it a good C3? And the answer is yes. Tony's done a lot of the photosynthetic work. He's done a ton of the physiology and gene expression work, and it's behaving just like every C3 we know about. So there's nothing remotely intermediate about it. We have a good chunk of the genome assembled, 502 megabases, giving us pretty much the gene space. And then he's compared the expression profiles to sorghum, maize, and rice. And by doing those comparisons, he's been able to reduce that number of relevant transcription factors to 8 from 118. So it's again, narrowing down the universe of the comparisons substantially. So if I can just summarize then this really brief introduction to the work that we've been doing comparing these genomes. We've been using phylogenetically informed comparisons, and that let us determine what aspects of C4 photosynthesis are necessary for the function of the pathway and separate those from ones that are simply incidental to very long evolutionary time. We've been able to reduce the number of transcription factors that might be of interest down to a very manageable number of 8, where they can start to be looked at individually. Found new gene candidates that are under selection, and we've been able to eliminate some other phenomena as perhaps being not relevant. This started out as a collaboration between my lab and Tom Bretnell's lab at the Danforth Plant Science Center. Since the project started, there have been 2 independent dispersal events. So James Schnabel has gone off to set up his own lab at the University of Nebraska, and Tony Studer is now at the University of Illinois, both still very much involved in the project. Thanks very much.
Other questions? I have one. The Dichanthelium is the C3 that's embedded within the other, or sister to one group of C4. Do you have any sense of what is that a reversion back to C3? Is there any sense in which that didn't quite make it and said, oh, I'll go back to being C3?
No, it looks like it's ancestrally C3. Mm-hmm. And what that little subset of the phylogeny I showed you, it leaves out an awful lot of other C3s.
Okay, so there's it's clearly separated.
It's clearly separated. Is that they're actually what I showed you is 2 independent C4 origins.
Great.
Yeah.
Thank you. One last question.
Yeah.
I'm just curious, did you see some of the structure of leaves variation between the species with close to maize? I mean the C4 and C3 and C4 types.
They're all NADP malic enzyme subtype, which is the subtype that has a single bundle sheath around the vasculature. So it seems that that single bundle sheath appears to have been derived both developmentally and phylogenetically from the meastome sheath, which characterizes all the grasses. So no, the ones that we are sampling are very, very much the same. The one that is different, and we have not investigated in detail, is switchgrass. That has a double sheath, but we haven't done the transcriptome work on that one.